training in Mysore, India, and graduated at the top of his university. He completed his psychiatric training in Bangalore, Vienna, London, and Detroit. He's received over 10 prestigious awards for his contributions to and excellence in psychiatry, clinical research, teaching, and mentorship. Dr. Keshavan is the editor-in-chief of Schizophrenia Research and serves on the editorial board for ACTA Neuropsychiatrica, Bipolar Disorder, and Asian Journal of Psychiatry. Dr. Keshavan's research focuses on the neurobiological basis of schizophrenia, especially as it pertains to first episode psychosis. His research on the neurobiology of psychosis, involving neuroimaging, biomarker, and early intervention techniques, has resulted in over 550 publications to date, including over 500 peer-reviewed papers and 4 books. Today, Dr. Keshavan will be discussing neurobiology of schizophrenia from soft science to hard science. So please welcome Dr. Matcheri Keshavan.
illness through the royal road of cognitive impairment, if you will. So let me tell you a little bit of a story about how my own interests got tweaked in this field. When I was beginning my psychiatry residency, I did some neurology, as I said, and my neurology professor didn't want me to go into psychiatry. And he said, Kesh, why don't you stay in neurology? And I said, no, I really want to. I went to medical school to become a psychiatrist. So he said, at least do some neurology research in psychiatry. So he asked me, and he was teaching, he was a dementia expert, he was teaching us how to do soft neurological science exams in the clinical bedside, clinical practice. And so he taught me how to do things like the graphesthesia or the grasp reflex, the palmomental reflex, and so forth. So he said, why don't you check it out in these functional psychosis? Soft neurological science should not be present in functional psychosis, and there should be a good way to distinguish between organic psychosis and functional psychosis. This is 1979. Those terms were still quite popular. So he taught me that a simple test to do in bedside clinical practice is the so-called palmomental reflex. So you scratch the palm of the hand with a sharp object like the key, and then you look at the face. It's like palmistry. Look at the hand and then look at the face. So you do that and you see a twitch of the chin when you scratch that. And that is a remnant of an ancient reflex, the palmomental reflex, which is a developmental reflex that is normally present at birth and disappears during infancy and the first couple of years of life. So this palmomental reflex, we hypothesized at that time, would be present or reappear in organic psychosis like dementias and post-head injury and so forth, but should be absent in schizophrenia. And we went and scratched the palms of about 100 schizophrenia patients and 100 controls and 100 organic psychosis in that mental hospital where I was working. And those days there was no IRB. We just took a notebook and went around and scratched the palms. And lo and behold, we found that the palmomental reflex was positive in 63% of schizophrenics and 65% of the organics and 13% of the healthy controls. So this was my first foray into schizophrenia research. So, that kind of really inspired my interest in understanding what happens to the prefrontal cortex in this illness, because these are frontal lobe release signs. So that brings us to the question, what do we know about schizophrenia and what do we not know? So there are a number of questions which I formulated in this talk. First of all, we know that psychosis begins in adolescence, but many aspects of this illness begin much earlier. So the question is really, when does the disease really begin? Secondly, we know cognitive and negative symptoms underlie the substantive disability. The question is, how are they caused? Thirdly, we know dopamine is altered. All our medicines work by blocking dopamine, but we talk about chemical imbalance. What is the chemical imbalance and where do we find that? And fourthly, we know the disease runs in families, but we still have only a beginning understanding as to what are the genes involved. And finally, we know that this is a highly heterogeneous illness and the disease boundaries are rather blurred. So is this one disease or many diseases? So these are the kinds of questions I have been focusing on over the last 25 plus years. So as I said earlier, the best way to understand our patients is to listen to their stories. And I do believe that symptoms and signs, even in psychiatry, are a very good way of understanding what's happening in that black box of the brain. So I put here a kind of a standard sort of a patient story, and we will come back again and again to this story to discuss the different questions that we raised for ourselves: when, how, what, and so forth. So this young woman, brought by her parents to the outpatient clinic, has some cognitive impairments in childhood, diagnosed as ADHD, has a family history of psychotic as well as bipolar disorders, begins to have her social withdrawal and declining grades, and some subtle, unusual ideas in adolescence, and then begins to have her first psychotic episode in college. As she is a freshman, she begins to hear voices, believes those voices are caused by a chip implanted in her head, and she assaults a classmate for this and gets hospitalized and is diagnosed with schizophrenia. And 4 years later, she continues to hear some chatter in her head and cannot work, mostly spends time in basement, little motivation, poor care of personal hygiene, has recurrent depressive episodes and manic episodes. And she's unconvinced about the need for medications and asks what is her illness really. So this is a standard patient you will see day in and day out, but they bring important questions to us. Why did my illness begin, in adolescence? What is the chemical imbalance I have? What causes my disability? And why did I get this illness? And do I have one disease or many? So let's take each question in turn. Why does this disease begin in adolescence? And it has been famously said that psychosis is actually a late stage of schizophrenia. There's a lot that has happened before that. We talked about in this case, there is the premorbid cognitive kind of underpinnings and a prodromal functional decline and cognitive decline and personality changes and subtle positive like symptoms, and then the psychotic episodes which are recurrent and they leave behind a persistent disability with cognitive and functional impairments. So these are the key facts of this illness, the premorbid, prodromal and psychotic phases of the illness, and then there is some post-illness onset functional decline in many individuals. So this is a slide from my dear friend and colleague Larry Seidman, who sad to say passed away about a month ago, a little over a month ago, and he contributed, in a phenomenal way to the understanding of cognition in this illness. And it's a meta-analysis, it's a combination of multiple meta-analyses, a paper we wrote a couple of years ago, in which he put together the degree of cognitive impairment in the premorbid and the prodromal and the psychotic and the chronic phases of the illness. What you see is about a half a standard deviation cognitive deficit in the premorbid phase and about 0.8 standard deviations in the prodromal phase in those who converge to psychosis, and then about a 1 standard deviation in the first episodes and about a little over that in the chronic patients. So there is evidence that there is a cognitive decline in this illness, and much of that decline happens between the premorbid to the prodromal period. So it's an important point to know in the context of early intervention efforts. So the question is, why is it that it is during this adolescence that cognition declines, and also it's during the adolescence that psychosis sets its foot? So there are a number of aspects that we know that we need to understand in order to know why psychosis begins in adolescence. Jay Geed, who is here, and I wrote a paper some years ago on why does psychosis begin in adolescence, and some of this work actually goes back to Jay Geed's work. And in Jay's work, it has been shown clearly that during the period of late childhood to early adolescence, there is a substantive decline in the amounts of gray matter, which might underlie the process of brain maturation during adolescence, which is what leads to this young girl to become the young lady later on. And that is a process that involves a programmed systematic elimination of synapses during the period of adolescence. Neurons are born at birth, they proliferate and network with each other, and then as time passes during adolescence, and also even during the intrauterine period, they migrate to the appropriate places, and then during adolescence there is the period of synaptic pruning, which then gives way to the process of myelination, which leads these neurons to become better insulated and better conducting circuits. And this is the way protracted development happens during adolescence. The idea that there is a synaptic pruning during adolescence, early adolescence and late childhood comes from the seminal work of Peter Huttenlocher, who showed that there is a substantive decline in the synapse density of brains of children and adolescents of about nearly 40%. And so that led to an important hypothesis about schizophrenia going back in the early 1980s by Irving Feinberg, not far from here, who said that some abnormality in this illness, in this process of synaptic pruning, may lead to too many or too few or the wrong synapses being eliminated. And he said, regrettably, we have no basis to choose among these possibilities. As a consequence of this bug in the genetic program, and he envisioned that this is a genetic program that might lead to the synaptic pruning process. This may lead to defects of neuronal integration, leading to the symptomatology of schizophrenia. So that was a very visionary statement at that point. So we read this paper. This paper, which was published in '82, had almost very few citations till the mid-1990s. So it was a remarkable paper. And we read this paper, and we did a review of literature at that point. And we wrote a paper revisiting the Feinberg paper in 1994, in which we looked at what all happens during normal adolescence, a reduction in gray matter density, reduction in synapse density, reduction in the prefrontal metabolic rate, reductions in the delta sleep, which is the distributed activity of multiple brain circuits during sleep, and also reductions in prefrontal membrane phospholipid turnover. All of these in normal adolescents. But if you look at schizophrenia, all of these are exaggerated. There is evidence for synapse loss, evidence for gray matter loss, evidence for hypometabolism in the prefrontal cortex, evidence for delta sleep deficits, which we had been looking at that time, and also reduced turnover of membrane phospholipids in the prefrontal cortex, which we were looking at through phosphorus magnetic resonance spectroscopy. So these observations led us to suggest that there is normally a synaptic pruning process that might happen during adolescence, but there might be an exaggeration of that process in those who are at risk for schizophrenia. So what might happen is the neurons that look like this in the middle during the fall or during the spring or summer of Boston seasons begins to look like the winter as it gets into the adolescence period of time. And indeed, David Lewis, in his landmark paper in 2000, showed that there is loss of synapses in the axons and the dendrites of postpartum brains of people with schizophrenia. Is what you see at the top are the healthy axons with the exuberant synapses, dendrites, and below are the axons of a patient with schizophrenia. So that brings us to another aspect of these connecting of these dots. So the question is, how does synaptic pruning happen after all? How does the brain know what to eliminate and what not to eliminate? We all know that there is long-term plasticity and neurons that fire together, wire together. And so obviously the brain must have a way of knowing what to eliminate and what is unnecessary and redundant, what is relevant and to be kept. So the one remarkable observation that was made not far from where I work at Harvard a few years ago by Beth Stevens' lab was that there are these glial cells which are normally inactive, but what do these glial cells do? They are basically phagocytes, and their job is often to fight external pathogens and so forth. But what about peacetime? the evolution has figured out an alternative job for them, and that is to scavenge the unnecessary, inactive, redundant synapses. When neurons are really not consistently firing, they come in and take away so that the brain is more efficient. Normally, there is a proliferation of synapses and network connectivity in childhood, but not all of that is necessary for an efficient functioning of the brain. And evolution has configured a way in which to keep the brain lean and mean by taking away the unnecessary synapses. And part of the job of these glial cells may be to phagocytose these unnecessary synapses, and they do so by tagging complement proteins. So which gives a eat me signal. So the inactive synapses which are not functioning because of the brain maturational processes, some are active and some are not, they are picked up and eaten up, if you will. So what Beth Stevens' lab showed, and this is a slide from her group, those red and blue dots there are the dendrites that have been eaten up by these phagocytes. In her experiments, she used the lateral geniculate nucleus, which receives visual inputs from both the eyes. The blue synapses are those that this phagocyte has engulfed, and the red ones are the opposite side, and so forth. So keep that in mind. We will come back to this process a little bit later in our effort to connect the dots. So there is another important observation that is being made, and that is the question as to how does the brain know when to stop pruning these synapses. So there is one line of thinking that there are molecular breaks that are built in that can tell us when it is time to stop pruning, and they might involve a sugar coating around the cellular and synaptic endings, and those are the perineuronal nets, and it's also genetically regulated. And so there is one idea that the synaptic pruning problem in schizophrenia, if it happens, may be related to an abnormal perineuronal net, perhaps due to this a breakdown, perhaps genetically related, of a failure of the synaptic, these molecular breaks due to glycoprotein problems. So that is the structure. We talked about what happens during adolescence in the brain, in brain structure. What about brain function? While there is a pruning of synapses, there is also an expansion of connectivity. So it's like when the small roads are going away in the woods, the highways are coming into place. So studies have shown that as time passes, white matter expands, white matter connectivity increases in the key brain circuits of frontolimbic connectivity and so forth, which may underlie the process of brain maturation. What about chemistry? David Lewis in Pittsburgh, with whom I worked for a long time, has done some really important work in this area. So the current understanding is that there are these excitatory neurons which are pruned out during adolescence, and then there are also another aspect of the neural circuitry that grows into the cortical regions during adolescence, which is also excitatory, and the kind of neurotransmitter system that's important for reward mechanisms, love, and risk-taking behavior, and so forth. And those of you who are neuroscience majors would immediately know what that is. That's dopamine. So what's happening during adolescence is there is still a lot of excitatory glutamatergic input, and there is also dopamine that is flowing in, which is what makes the brain highly hyperexcitable. But evolution has it that there are brakes as well as accelerators during this period. So there is the GABA system that also matures during this period, which is why teenagers are normally, most of them are not psychotic, because there is this fine tuning of excitatory and inhibitory circuits that gets established during adolescence. So David Lewis has written extensively on this functioning of the circuitries of pyramidal neurons and the local circuit neurons like the parabolic positive neurons, the chandelier neurons and so forth, which are GABAergically mediated. And there is the circuit with glutamate, dopamine and GABA that really sets the stage for a mature brain. So in summary up to this point, during puberty, the brain stops automatically adding new connections and starts pruning away necessary synapses. The frontal lobes are still forming during this time and still becoming more efficient at conducting signals. And the adolescent brain is at the peak of its learning ability and is not fully able to inhibit impulses. Young man, go to your room and stay until your frontal lobes finish forming. And so this is really the kind of idea behind the paper Jay and I wrote on why adolescence is when psychiatric disorders are at their most maximum. But what happens in those at risk for schizophrenia and those who have developed schizophrenia? For many years, we have been following a group of familial high-risk offspring in which we have seen that the degree of gray matter reduction in these familial high-risk offspring is steeper in these kids compared to their healthy, typically developing adolescent comparison subjects. And the same kind of thing is what you see in the prodromal period, and a number of studies including Trey Cannon's paper, Kristen Caden is here. The Naples group has shown that there is a gray matter loss that is steeper in those at clinical high risk who are converting to psychosis compared to those who don't convert to psychosis. So if you move forward towards the early course of schizophrenia, you see that this gray matter loss is continuing probably in the first few years of the illness. This is Paul Thompson's paper going back several years, showing that there is this continuing decline in the first few years of the illness. Sorry about that. So a number of studies, including our own group, have shown that in the first psychotic episode, and this is a cohort of never medicated first episode patients, we have seen that they have substantive cognitive impairment, as you see on the top right, with the bottom line being the first episode schizophrenia patients in yellow, cognitive impairments across a wide range of functions. And then on the left, what you see are soft neurological signs abnormalities, which is what I started off talking to you about. Very robustly increased in first episode psychosis patients, and then gray matter reductions in first episode psychosis patients, as well as prefrontal evidence of prefrontal membrane phospholipid declines. So let's come back to our patient. So the question this patient asks is, she doesn't think she has any illness. Everybody talks about her having a chemical imbalance. What exactly is this chemical imbalance? So that brings us to the question, going back to the 70s and 80s, as I learned as a medical student, was that too much dopamine is what causes schizophrenia, and that was a simple, straightforward story. But then as I was doing my residency and later it became clear that it is not as simple as too much dopamine, Danny Weinberger proposed this elegant theory that what might be happening is that there are two kinds of problems. One is that there is perhaps a dopaminergic excess, and in the mesolimbic dopamine tracts, and then there might be a dopaminergic So what is missing here in this, yeah, okay. So the next slide shows that evidence for the idea that there may be an elevated dopaminergic function in schizophrenia. And studies have indeed shown that there may be some increase in the dopamine receptor availability, which may be largely an antipsychotic drug effect. And also, if you look at dopamine transporter availability, there is not much of a difference. So many times, for many years, people thought that the dopamine PET scan studies are really not showing much. But in the last few years, there has been an increasingly consistent observation that in untreated patients, there is an increase in presynaptic dopamine synthesis, as may be measured by fluorodopa PET studies. This is work by Oliver House at the Maudsley in England and so forth. So there is some evidence that there is dopaminergic excess, perhaps presynaptic. But then Danny Weinberger proposed this elegant theory that it may not be just dopamine. It may be that while these psychotic symptoms are explained by excessive ventral striatal dopamine, it doesn't explain why people have negative symptoms and cognitive impairment. So he proposed that there is a mesocortical dopamine dysfunction, which may explain cognitive and negative symptoms. So that was an elegant theory, because at one stretch he could explain all aspects of schizophrenia. But the problem is, why do people have two different kinds of circuit problems? One mesocortical and the other mesolimbic. So that brings us to the question of what is it that regulates limbic dopamine, and what is the reciprocal relationship between cortical dopamine and limbic dopamine. And that is where another neurotransmitter comes into the picture, and that is glutamate. Glutamate is the corticofugal abundant neuronal circuit that is involved in regulation of limbic dopaminergic tracts. So that raised the question as to whether schizophrenia is related to more than just dopamine, perhaps related to a dysfunction in the glutamatergic underpinnings, which may lead to a decreased tonic glutamatergic function, and perhaps related to NMDA receptor hypofunction, which may lead to an inefficient overproduction of glutamate during periods of acute psychosis. Something we proposed and many others in the 1990s. And indeed, some of the studies in recent years have suggested that there is indeed a glutamatergic problem from spectroscopy studies. What you see is a meta-analysis at the top, where a number of studies do show that there is evidence of increased glutamatergic levels, the glutamate-glutamine ratios, and we also observed that in those who are at familial high risk for schizophrenia, especially if they have high levels of schizotyping. So the chemical soup of schizophrenia might be a combination of a glutamatergic and a dopaminergic problem. But then it may not be even as simple as that. It could be more than dopamine and glutamate. And there is this yin and yang theory. We know that glutamate is the yin and the GABA is the yang. And so whether there is a glutamate-GABA balance that is the problem is another question. And there is beginning data to suggest that there may be a GABAergic problem in this illness. This is a study from Pittsburgh, published a couple of years ago, showing that the flumazenil binding after tiagabine is increased in unmedicated patients with schizophrenia, as well as the never medicated patients. Small study to be replicated, but suggests that there may be primary GABAergic problem as well. So to put these together, the current beginning thinking is that there may be a glutamatergic problem, perhaps relating to NMDA receptor hypofunction, which may lead to inefficient glutamatergic activity, which in turn might lead to a excessive unregulated mesolimbic dopaminergic activity leading to psychosis, and an inefficient mesocortical dopaminergic activity leading to cognitive and negative symptoms. So that's kind of the chemical imbalance of this illness. So moving forward, the next question she asks us, this patient, is she has a strong family history of schizophrenia and bipolar disorder as well. And what is it that I'm inheriting that might be causing my illness? So that raises the question as to another set of emerging observations that can help us connect the dots between the biology and the symptoms and the science and the synapses. So this is the Manhattan plot, as you see here. There is the Statue of Liberty there, and the various skyscrapers. These are the, you know, most of you are familiar with the Manhattan plots, where if these skyscrapers go above the p to the power of h or higher, they are significant at a genome-wide level. The x-axis here is the genome, and the y-axis is the significance level. And as the sample sizes have increased, the number of hits for schizophrenia has progressively increased from just a handful to most recently about 108 or so, and still growing. So one interesting peak among all of these skyscrapers in this Manhattan plot, which has been there for the last several, probably a couple of decades, is on the chromosome 6p, which is the region for some major histocompatibility genes. So suggesting that there may be something immune that's happening. And interestingly, very recently, in the last couple of years, it was observed that this particular area is rich for a set of genes that relate to complement proteins. So here is a dot again being connected. So we talked about synapses and the synaptic pruning and the repurposing of complement proteins to normally eliminate these synapses. And now these same complement proteins appear to be showing up in the genes here. So this is work by Steve McCarroll. Saker, who was a medical student at that time at Harvard, was the first author on this Nature paper. Interestingly also, these studies are beginning to show that the glutamatergic and neurodevelopmental genes are among the majority of those gene locations that have been observed. So Steve McCarroll went on to observe that the C4A expression was increased in postmortem brains of schizophrenic patients, and the C4A copy numbers actually correlated with the degree of schizophrenia risk. So there are multiple lines of evidence suggesting that these immune protein genes may be involved in some significant way. We also have been looking at the plasma complement C4A levels in first episode schizophrenia and CSF in unmedicated chronic schizophrenia patients, and we have some suggestive evidence that C4 might be increased and also fairly strongly correlated with levels of complement CRP levels, which is an immune modulator. But you know, the humbling part here is that the complement genes may explain only a very small proportion of the genetic diathesis for this illness, and a collection of multiple genes that are related to synaptic integrity and neuronal adhesion and neuronal migration processes might contribute collectively to schizophrenia. It would be wrong to say that, you know, the problem of schizophrenia has been solved through these immune gene discoveries. Going back to our patient, she is now, you know, several years later, still quite ill, functionally impaired, and you know, not working. And so the question is, what is it that might cause this kind of functional disability in this illness? So probably it is good to think of schizophrenia not as one single problem, but it is a result of a series or a cascade of sequential hits that the brain takes. And at the pathophysiological level, there might be an impaired neuronal formation, neuronal development in the intrauterine period and early development, and then a failure of brain reorganization during adolescence, and finally also a series of problems on brain preservation as we go into the more chronic phases of this illness. The various processes being involved might involve synaptogenesis, neurogenesis, migration, neuronal pruning, circuit plasticity, myelination, as well as glial support, cerebral housekeeping, myelin repair, all of these processes might be at work. But the disease is probably very heterogeneous, both longitudinally and cross-sectionally, and all of these different processes might be involved at some point or the other. And one might increase the chances for the other process. And genetic factors are clearly important, but then they don't act by themselves and probably interact with multiple environmental risk factors, which may include the phenomena such as infections, trauma, hypoxia, winter birth, urbanicity, malnutrition, and so forth, early in development, substance abuse, psychosocial stress, social isolation, and so forth during adolescence, and subsequently, even after the illness has set in, the process of oxidative stress, neuroinflammation, substance abuse, medication effects. We don't know that. Well, maybe some antipsychotic effects might contribute to the pathology as in addition to being helpful. And oxidative stress might be an important process which we only have a beginning understanding. There are these pro-oxidant systems and the antioxidant systems. The pro-oxidant systems, involve free radicals, which might damage the neurons and the mitochondria, which in turn might compound the problem. Smoking and inflammation and the diet and viruses, they all might have a role to play. And we were talking yesterday evening, the microbiome may have a significant role to play as well in the pathogenesis of this illness, but may also be a consequence of the lifestyle changes with this illness. So we go back to the elephant that we started off with. So what is this elephant? What do we see? What causes schizophrenia? There may be multiple processes which may be related to each other, that may involve multiple neurotransmitter systems, dopamine, glutamate, and GABA, and multiple processes such as inflammation and oxidative stress and hormonal imbalance, related to genetic and environmental factors and working via epigenetic factors, leading to the complex phenotype which we call schizophrenia. And all of these may be related to a common pathophysiology with multiple interconnecting nodes, and so it might all be one elephant, you see here, or it may be multiple. This cartoon I put in here. It's a very simplistic model. If only it were so simple, but it's probably not so simple, and we'll come back to that point a bit later. So now our patient says she also has been diagnosed with schizophrenia this year. She was diagnosed as bipolar a couple of years ago, schizoaffective at another time, and so on. So she asks me the question, my diagnosis keeps changing, and do I have one disease or many? So what is it? So that's the question that we have been trying to address. And so that is a central question in a consortium that we have called the BSNP, that is Bipolar Schizophrenia Network for Intermediate Phenotypes. And there we have taken an agnostic approach to this illness, arguing that the Kraepelinian boundary of schizophrenia and bipolar disorder may be a problem, and that there may be different diseases within this spectrum that do not read the Kraepelinian boundaries or do not map onto the DSM. So with that in mind, we developed a battery of biomarker tests based on cognition and electrophysiology that included a number of measures that map onto either cognitive control or sensory motor reactivity based on EEG and ERPs and cognition. And we ran that battery agnostically across about 2,000 subjects with schizophrenia and their family members. And we, using a machine learning kind of approach, K-means clustering, we looked at whether there are subgroups that fall out of this and might map onto DSM or may not. And what indeed we found was that there are three subgroups. We call them biotypes. And biotype 1, you will see later, was the one that had the most brain abnormalities, and with gray matter losses and so forth. Biotype 2 was the one with, and the biotype 1 had a lot of cognitive impairment. Biotype 2 had moderate degrees of cognitive impairment and had many electrophysiological signatures that were kind of the opposite of biotype 1, like exaggerated amplitudes of ERPs and increased spontaneous amplitudes of resting state gamma and so forth. And biotype 3 was very interesting in that it did not differ from healthy controls. And they had substantively high proportions of cannabis histories. That's why it's in green there. And family histories of psychosis were most prominent in biotype 1. So that made us wonder whether there might be etiologically distinct subgroups within this psychotic spectrum. So, whenever we do something like this, we need to validate. So we took biological measures that we did not use for the construction of these biotypes, and then looked at how well these groups separate. And first of all, this is Brett Clements, who wrote that paper last year on biotypes. And the different colors there are the different diagnostic groups, red being schizophrenia, green is schizoaffective, blue is red being the bipolars, green is schizoaffectives, and the blue are the psychotic bipolar disorders. So all these biotypes are approximately similarly distributed across the 3 DSM categories. So basically, this classification did not map onto DSM. It was kind of orthogonal to DSM. So if we looked at the gray matter densities, biotype 1 was showed maximum and diffuse gray matter abnormalities. Biotype 2 had more focal anterior limbic abnormalities, and biotype 3 did not show differences from controls. Now, if you look at first degree family relatives of biotype 1, they look like biotype 1. First degree relatives of biotype 2 kind of look like biotype 2. And first degree relatives of biotype 3 kind of look like biotype 3. So this was one level of validation. And the other thing that we did was Cynthia Guillemont, one of our postdocs in my lab, she looked at the hippocampal shape across these diagnoses, and she found that, irrespective of whether you call them schizophrenia, schizoaffective, or psychotic bipolar disorder, there are hippocampal abnormalities across the groups. But if you look at biotypes, you see that biotype 1 is the one that shows the maximum amount of hippocampal loss, as well as hippocampal concavities in the anterior hippocampus. So another postdoc in our group, Sinead Kelly, she looked at the fractional anisotropy levels in the corpus callosum across these groups, and she again found that biotype 1 has the maximum abnormality compared to the other groups. So what we are seeing is that these biomarkers, which were originally not used in the biotype construction, were separating themselves better across the biological subtypes than across the DSM subtypes. So it may be that there are many diseases, and this is back to that elephant. It may be that there are multiple elephants that you see, or maybe even multiple mice, so to speak. So what you need is animal models that take discrete aspects of this illness and construct models, as opposed to any single model, which is almost impossible for a complex disease like schizophrenia. It also leads to the question that can we identify and develop better treatments if we stratify this illness into biologically more meaningful subtypes, and then test compounds or new treatments in those kind of subgroups. So, and fortunately now there are many ways of targeting the synapse, multiple synapses, whether it's glutamatergic, GABA, nicotininergic, dopamine, cannabinoid and antioxidant systems, anti-inflammatory systems, and so forth. It may be that there are multiple pathophysiological processes in the same individual, or multiple subgroups of this spectrum with one or other pathophysiological process going array. And so going forward, we would need to identify subpopulations based on biology and test novel treatments using those biological markers as proxy outcome measures. So this is what leads us to this experimental medicine approach articulated by the NIMH. Complex slide. So also pharmacology is not the only way, and there is a lot that we can do to improve the brain simply by exercising the brain. So we have shown that it is possible to change the brain structural integrity by cognitive enhancement therapies. This is Sean Eek in our group, now in Pittsburgh, who showed that hippocampal volume and cingulate and the insula can actually be and the parahippocampal gyrus fusiform and the amygdala can actually be increased in people following treatment with cognitive enhancement therapy and approach to cognitive remediation. So prevention is possible at several stages, and one can think of prevention in terms of primary, secondary, and tertiary. If one considers this illness as a complex series of or a cascade of events that begins at birth, there is possibly many things that we could do at birth, and then subsequently early detection, parental support, family-centered care, CBT, anti-inflammatory treatments, antioxidants, omega-3 fatty acids, GABA modulators, and then going into the more chronic phases, preventing relapses by optimal psychotherapy, CBT, exercise, supportive employment. All of those have a place in this overall scheme of things. Prediction is equally important. Now the knowledge base is expanding enough to be able to possibly predict psychosis with reasonable degrees of accuracy. This is on the left is the Naples work showing that the area under the curve for prediction of psychosis in clinical high-risk individuals is reasonably good, like a 75% or so area under the curve. And we have done a similar kind of a calculator using environmental risk factors in the familial high-risk population. Jaya Padmanabhan in my group has done that. And we find similar predictive ability using a simple calculator. So my key points are, firstly, that schizophrenia develops perhaps from an aberration in the synaptic refinement process during adolescence and a failure to fine-tune excitatory and inhibitory circuits during a critical period. But the question of what are the brain changes that underlie these core deficits, structural and functional deficits in synapses may underlie these developmental processes. Dopamine may be proximally involved, but may not explain everything. But imbalances in glutamate and GABA and other molecules that underlie synaptic function and brain development are involved as well. Going to the question of why psychotic disorders are likely to result from polygenic and poly-environmental risk factors and their interactions. Certainly, they are those genes that affect brain development, synaptic integrity, immune and channel function. What we define schizophrenia as by DSM is probably not one disease. The developmental brain abnormalities in this illness may non-specifically cut across many symptom-based diagnoses, and there may be distinct biological subtypes of psychotic illness. Where do we go from here? The emergent observations set the stage for personalized prediction as well as targeted interventions in populations stratified by neurobiology. I think that's my last slide. There are many people that have worked with me, both in Pittsburgh as well as in Boston, and those are my funding agencies. My only disclosure is that I edit schizophrenia research. And so with that, thank you all very much. Appreciate your interest.
So while we're getting the microphone going here, good. Kesh, I want to thank you for this tour de force from frontal release signs to the much more complex biology of the schizophrenias. I like our trainees in the room to take note that what you saw here, really a comprehensive description of the biology of a complex phenotype, started with a clinical observation at the bedside with the palmomental reflex. I think many of us started there, and that may not be the place that you start today. Maybe today you're starting from the last slide you saw that Kesh put up, and as the course of your career evolves, it will look as different as this does to our palmomental reflex, and that's the process of studying the biology of complicated diseases. So thank you, Kesh, for taking us through that pathway. Questions for Dr. Keshavan?
Yeah, thanks for that very comprehensive presentation. Clinically, given that women have a later onset of schizophrenia, a more benign course, but are at increased risk for late onsets, and that estradiol can affect dopamine and progesterone and GABA systems, and they both can affect immune functioning and synaptic plasticity. Could you comment on the role of gonadal hormone change in the course of schizophrenia?
So that's a very important question as to why women have a later onset, have a more benign illness, and tend to do better over the course of time. And there may be developmental differences, and probably Jay Gid can shed more light on that question. He has wrestled with that a lot. But many years ago, we did a study published in the early 1990s, looking at that time a large sample of healthy youngsters whose brains we had looked at as a function of age, and we saw that the steepness with which gray matter declines during adolescence was much steeper in the boys compared to the girls. So there may be a protection against a rapid synaptic pruning process in girls, and that could be estrogen related. Could be, that could be one factor, and there may be some genetic and hormonal protection against synapse loss in the girls. And secondly, it's also possible that estrogen has its effects on dopamine, and whether it can kind of blunt the onslaught of dopaminergic excess by being an endogenous dopamine antagonist. So that's another question that has been raised, but Jay may have more to say. Yep. Yep. Yeah.
Kesh, were there sex differences in your three biotypes?
There were no sex differences in the three biotypes. There may be a slight excess of men in the biotype 1, but it was not statistically significant.
Kesh, thank you for a very comprehensive presentation. One key point which you from the beginning and to the end is the synaptic pruning. Do we have any good evidence of this pruning has anything to do with the genetics component? Have you ever looked at the familial schizophrenia versus non-familial? What is the synaptic pruning? In what way is different? Or you may have some other postulate to see the synaptic pruning and the mechanism of synaptic pruning, why there should be synaptic pruning and leading to abnormal mental state. This is a very important key point of which I learned from you.
So these are very, very important questions. So, what is the genetic basis of synaptic pruning? We do know that many genes are involved in that synaptic refinement process. And one example which I showed here is the functioning of the complement proteins, which may regulate the timing of when synaptic pruning happens during adolescence. And the other example I showed you is in terms of the perineuronal nets, and they may be involved in the time of offset of synaptic pruning. When would pruning end and when would pruning begin, and so on. They both might be related to pathophysiological or neurodevelopmental processes which are under genetic control. And the example I showed of the complement protein is that now there is the connection between the gene implied, which is the complement genes, and the process, the pathophysiological process. And some studies have shown that even the perineuronal net related proteins might also have a genetic linkage to schizophrenia. So the evidence is beginning to be indirect, but there is a suggestion that familial factors are involved. So genetic factors are involved in the synaptic pruning process. But there are many more questions to answer. For example, do we have a knockout mouse in which pruning is exaggerated? In fact, Steve McCarroll's lab has done some of that. If you knock out C4A protein, you do see some of these cellular phenotypes of schizophrenia and so on. So I think there is a lot more questions to be answered, but it appears that genetic factors are involved. But we only know a little bit. There may be many other genes involved. There may be many other environmental factors that are involved as well. Hormonal effects clearly might impact on the rate of synaptic pruning and myelination process and so on. And pruning may be only one process. There may be many other processes. Myelination itself, oxidative stress. Yeah.
Wonderful talk. In terms of the biotypes, I understand that the BSNP sample ranged in age. Is there anything that you can say about the development of the different biotypes? Are these things that you think have been there all along, or do they represent kind of in stages of different paths of development? And one of the reasons I ask about that is because, correct me if I'm wrong, but it looked like that maybe most of the slides, biotype 3, basically looked like healthy controls, but in your postdoc's hippocampal shape analysis, it looked like that they were kind of super healthy, or that their hippocampus was sort of different from controls in the opposite way that biotype 1 was. So do you have any sense of, are they kind of a resilient group, or are they a compensatory group? Or
yeah, so about your first question, whether they are different in their age, we did not see that they were any different in their age. And secondly, whether they are a beginning stage or a chronic stage and so forth. We only have a cross-sectional sample of people at multiple ages, and we don't have longitudinal data yet. We just got a small supplement to this grant to follow up these people and establish the stability of biotypes. We also have another study that's midway in McLean, in first episode patients, to biotype them and then follow them up to see how they are doing 2 years and 3 years down the road. So that's happening. That is a very interesting point that you raised as to whether biotype 3 is showing some phenomena that are opposite of what biotype 1 show. It's quite possible, and I would say likely, because there may be compensatory processes that are happening in some of these individuals to an underlying state of vulnerability. And some of these may take the form of aberrant neuronal excessive plasticity in some brain region. It's quite possible.
I have a question about the neurocognitive enhancement you mentioned.
Yeah.
What is the age range of the people who showed like an effect?
Yeah, so the age range in that group was between 18 to 35.
Okay, and no studies about like later in life, or how?
There was a study prior to this study that I showed. Jerry Hogarty published a paper using the same methodology, cognitive enhancement therapy, in chronic schizophrenia patients, where the mean age was around 45.
Last question, what kind of therapy was that? What kind of enhancement was that?
So this is a comprehensive, stepwise cognitive remediation that takes cognition as well as social cognition in a stepwise manner. So we do computer-based cognitive remediation for a few months, followed by individual and group-based social cognitive exercises.
Hi, Dr. Keshavan. Thanks for the talk and trying to connect all the dots. It was really useful as a student. So my question's about, so in the beginning of your talk, you sort of talked about excess synaptic pruning and all the evidence for that. And then you transitioned into the neurochemistry and talked about kind of glutamatergic hypofunction, dopaminergic overactivity. So my question is, has there been any evidence, or has it been kind of rigorously shown that in the synaptic pruning that there's like a differential effect on some of those circuits, or is that still an open question, or is it sort of just a global synaptic pruning?
Yeah, so the neuropathological studies that have looked at synapse loss in schizophrenia have mostly looked at the pyramidal neurons, which are glutamatergic. So the excessive pruning that is hypothesized in this illness might largely affect these glutamatergic synapses. So that might suggest that the fundamental problem might be glutamatergic, but we don't know the answer to that yet. And the dopamine problem might be a consequence as opposed to being the primary problem. But on the other hand, it's also possible that the fundamental problem may be GABAergic, which leads to an abnormal refinement of glutamatergic neurons during adolescence. We don't know what comes first and what later. But the pruning might happen largely in the overabundant synaptic machinery in the brain, which is glutamatergic.
Thanks for a great talk, very lucid. I'm curious to know what you think about neurodegeneration in schizophrenia after the first few years. I'm not convinced there is, because if you follow these people longitudinally, they do not become more, they're not more likely to become demented. They die young, but they die of heart disease, cancer, not dementia. They don't develop dementia early or even late for that matter.
Right.
Even when they die at 78, the brains don't look like those of people with dementia. So I'm not sure it is.
Well, I agree with you that degeneration may not be a common problem in this illness. Studies that have looked at longitudinal course of schizophrenia, like Jean Rund in Scandinavia, has shown that cognition remains about the same through the course of the illness. But there's one problem with that. There is heterogeneity. If you look at the subgroups of schizophrenia, there are some who bounce back and actually get better than their early course of the illness. Some who go down and the others remain in the middle. So taking a large group and showing no change doesn't make much sense. There may be a subgroup that does show further decline, and I think that when it does that, when that happens, that may be related to the disuse atrophy of the brains of these people, because chronic illness, negative symptoms, not using their brain might lead to a certain degree of cortical thickness loss. And I am increasingly believing that there is a small but significant effect of antipsychotics too, which may lead to some cortical atrophy. We need to understand that better. So there is probably some decline, but it is not the kind you will see in dementia. And probably not a degenerative process, maybe a disuse atrophy process.
So we have time for, it looks like two more quick questions, and then we're going to wrap things up here.
So I recently was attending the National Network of Depression Centers in a presentation on depression similar to yours, a comprehensive approach. And I have to, this is the way to do research, to combine clinical and mechanisms. But I'm wondering, given the current climate where you can't do mechanism studies and have clinical outcomes, and you can only have one specific aim, that's which your power analysis is based. How do you propose going forward with this? I mean, do you submit your grants one particular, pathogenic system per grant, or what's your? I'm just curious. I would love for the, I was sitting next to one of the program officers who said, this would never be funded.
Yeah.
The extra.
Well, we're all trying to figure out how to adapt ourselves to the rapidly changing system. And I don't have a very good answer to your question. I think we have to set our sails to the way the winds are blowing, but then try to do our answer our questions the best way possible. And I would say there are ways of isolating questions into single, simple constructs and testing them, and that is probably a good way to go. The days when we could do, 1,000 subject study for 100 years in a large sample is probably not going to be that easy. But on the other hand, the current day and age is also increasing the ability to do multi-site studies and big science and connecting up and harmonizing large data sets and answering the same kind of questions that we used to do in single sites before. So there are some downsides to what's happening, but there are also some upsides. So I would say, to my mentees, what I say is try to get into big consortia, big groups, and try to get access to existing data sets, and you may be able to answer more questions that way.
Eugen Bleuler wrote about schizophrenia 100 years ago, and he described a lot of patients with catatonic schizophrenia, which you don't hear much about. And it just makes me wonder how much you can compare a 20-year-old schizophrenic today to an 80-year-old schizophrenic today in terms of origins, and maybe autoimmune origins being more of an issue now versus 50 years ago. So how much do you think you can compare across generations?
Yeah, so I think it's an important point you bring up as to whether we used to see a lot more catatonia before and not as much now. I think my answer to that is that I do go on ward rounds and see patients in the acute inpatient unit. I still see catatonia, a significant number. So it may be a little less than before. Probably the way they present dramatically these days with catatonic posturing and so forth is because of increasing early detection and prodromal intervention and so on. It may not necessarily be that the disease is changing its etiology or pathology and so forth. But on the other hand, it's also possible that there are some changes that we are not fully understanding. It may be that there are changes in lifestyle, the way we eat and the gut microbiome and how that is affecting inflammatory processes and allergies, and we have no idea. And how the environmental pollution might be contributing, we don't know. Big question.
Still many unanswered questions for our trainees to pursue in their productive careers of schizophrenia research or whatever disorder you tend to find. Kesh, I would like to thank you on behalf of the department. We have a tradition for visitors or grand rounds. We hope you'll be returning to San Diego frequently, visit family, visit the new family here in the department. Thank you for the time you spent with us and for the outstanding grand rounds.